Everyone, welcome on YourPedia Media, and today with me are Parvez Bakht and Ma'am Rinki Gupta. Welcome both of you. So both of them are research scholars from IIT Roorkee. Both of you represent molecular bacteriology and chemical genetic lab, and you gave a presentation, which is a wonderful presentation on methods and composition for identification of novel anti-persister activity of GRAS compounds. Now, I would request Rinki you to first tell our viewers very concise way what this product is all about.
Yeah. So in brief, basically what we are trying to do here is, so we are targeting anti-persister. We are using anti-persister molecules which are generally regarded as safe compounds against bacterial persistence. So the major problem that we are trying to tackle in here is antimicrobial resistance, which I am sure most of the public is aware of. So it has been estimated that by 2050 there would be like 10 million deaths which would be attributed to AMR, like antimicrobial resistance, which also includes antibiotic resistance. So we have seen that recently after the COVID pandemic, it was also labeled as the silent pandemic, because it is silently taking up all the lives. It is going to surpass cancer patients, like in terms of its mortality rates, in terms of its cases of evidences or incidences as well. So AMR, one of the major reason of development of AMR is also bacterial persistence. So bacterial persistence is what is, it's basically generation of dormant bacteria, which under the like pressure of high concentration of antibiotics, they become dormant by developing certain kinds of mechanism inside them. And they lead to recurrent infections. So you must have heard in clinics that after a certain period of antibiotic treatment, the infection relapsed. That is because of the generation of bacterial persistence. So in here, what we have done is we generated. So we used basically carbapenem resistant A. baumannii pathogen, which is a WHO deemed priority pathogen. It basically comes under the critical priority pathogen list. So where WHO has recommended every scientist working on this aspect to to look forward the mechanisms or treatment options to be developed against this pathogen. So what we did is we used a carbapenem antibiotic, which is a meropenem, and this is a last resort antibiotic for the treatment of A. baumannii. And we have seen increasing incidences of development of resistance in this pathogen against these last resort antibiotic. So one of the mechanism is just like I said, it's bacterial persistence. So we developed persisters of A. baumannii against this antibiotic, and we tried to screen molecules which are generally regarded as safe. So these were basically essential oils, and we fortunately we found that thymol, which is an essential oil, and it was able to kill these persister cells. It was able to eradicate all of them, and the good thing about that is it is basically a broad spectrum molecule. So we also tested it using other antibiotics. So like what happens is, generally you can use other antibiotics also for the treatment, and we see the development of resistance against other antibiotics also. So we saw that if we are using, like for example, rifampicin or tigecycline, other antibiotics, we generate persisters against those antibiotics also. This thymol is able to eradicate those persisters also. So it shows broad spectrum activity in terms of the antibiotic which is used. Also it shows activity in terms of what kind of pathogen you are using. So there are other pathogens like serious pathogens, pseudomonas, which is known to cause pneumonia, and there is Klebsiella pneumonia also. So these pathogens also affect the lungs. So we see that the thymol is able to kill the persisters of these pathogens also.
Wonderful.
Further, we have in our lab, we have collaboration with like hospitals all over the India. So we have obtained like some clinical isolates which are isolated in hospitals from patients. So we procure those clinical strains, and we tested this theory against them as well. So we saw that these clinical strains were also able to develop these persisters against meropenem, and thymol was able to kill it. Now we also did what we did is we used a mechanism based library. So mechanism based screening. So we see that why thymol is able to kill this, because the mechanism which they use to generate these persisters, it is able to nullify that mechanism itself. Like what these persisters do is they will efflux out, they will push out all this antibiotic outside of it, so that it is not able to target the basically the proteins of
Okay.
bacteria. So this thymol, what will it do? It will inhibit this efflux pump.
Okay.
So that antibiotic gets accumulated inside the cell and it is able to kill it. So in this way, it also shows combination therapy with meropenem itself.
Okay.
Because now it has inhibited the mechanism of persistence. So meropenem again is able to kill it. So this way, the combination is also very efficacious against this.
Okay.
So the key points of this technology for us is it is a generally regarded as safe compound. So it both meropenem and thymol, these are FDA approved drugs. So they are also available in market. So we have just identified a new potential application of this compound, and also it is a broad spectrum anti-persister molecule, and there are no existing therapy till date which are known to have anti-persister activity. So this way, and our technology is currently at TRL 3 level.
Okay.
And we plan to move ahead to do some other preclinical evaluation.
Okay.
So till now we have used a wound infection model on biopsy mice, and we have seen good efficacy of this combination on wound infections caused by Ebommeni infections. But we also plan to do it with other types of infections, like for example systemic infection, which is really challenging to develop with persisters.
Wonderful. Okay, so Rinki, wonderfully explained, though I'm totally illiterate in this field, but wonderfully explained. So my next question to you is, come to the point, you are here for collaborating with the industries.
Yes.
So Rinki, very straight question to you, where do you find industry gap? Anyone of you can answer. Where do you find industry gap? What I mean by that is कि इंडस्ट्री वो प्रोडक्ट मार्केट में लेके आएगी जो प्रोडक्ट की रिक्वायरमेंट है मार्केट में. सो इफ यू फाइंड सम काइंड ऑफ गैप इन दैट वे, आई मीन बिजनेस मॉडल बनता है इफ देयर इज अ नीड, you have a product and you are the best in the product and there is a scalability existing. So where do you see that?
So like I said that it is a major problem in health settings, in healthcare settings. AMR is a big challenge. So to counter that, obviously we need help because we need to commercialize this combination as anti-persister therapy. And also before doing that, so for regulatory approvals or other things, we need to do the clinical trials. So that is the major gap, I believe. So where we would like to have the industrial collaboration with us, so that we can move it from the preclinical.
And why industry will do that? What is the benefit to the industries in future?
So like I said,
they want to get assured actually that your product we can sell.
Yeah.
So what is your assessment on that?
So like I'm saying that AMR is a big like significant threat to public health. Like people are dying. Obviously we want to give them some hope to. We don't want to have that hopeless time where they will see that none of the antibiotics are going to work. And this was situation. It was close to me when I have seen one of my acquaintance. They were not able to get treated by any of the antibiotics. They had some different kind of treatment for some other disease, and just because antibiotics did not work on him, and he just died.
Okay.
So this is one of the post antibiotic era that we are kind of entering into. So this will lead to failure of chemotherapies, because if we do not have antibiotics to treat these kinds of infections. So
okay, Parvez, a question to you, fast question. क्या ऐसा कोई प्रोडक्ट ऑलरेडी मार्केट में है? डू वी हैव कंपटीटर्स?
या लाइक मार्केट में अभी तक तो प्रोडक्ट ऐसा नहीं है, लेकिन बट पीपल हैव ट्राइड, लाइक दे हैव डेवलप्ड सम कॉम्बिनेशन एज वेल एज सम एलोन थेरेपी. सो उसमें ऐसा है कि कुछ चीज़ें अभी रिपोर्ट की गई है, बट दे आर आल्सो ट्राइंग. सो एक्चुअली अगेंस्ट एज ही सेड लाइक कार्बापेनिम रेजिस्टेंस, उसके अगेंस्ट अभी कोई थेरेपी अवेलेबल नहीं है. सो दैट्स व्हाई वी आर प्लीज लाइक यू कैन ट्राई योर बेस्ट to collaborate or like convince people from industry so that they can collaborate and they can look into very detailed and at ground level what exactly we have done so far, so that they can
so in this entire supply chain, physician doctors are also one of the stakeholders. I'm sure you have gone to technology readiness level 3, I suppose. You must have done some assessment with the physician and doctors also. What is their take? What do they say about this?
So
if you have
no, actually we have all done this all at the laboratory level only.
Okay.
But we have procured the clinical isolates from hospitals.
Mm-hmm.
So we have tested it on them. So yes.
Okay, so I think if you can also tell me, like here on the basis of the innovative research which you have in the lab, either you can collaborate with the industry or the other stakeholders can be the entrepreneurs, budding entrepreneurs like you people only, who can start up around your research. Do you also see opportunity for them? And if yes, what is the kind of capital investment or funding required for them to create something out of it? If you have thought of it, if there is a scope for entrepreneurs.
So I think what I believe is the problem that we are tackling is is basically the healthcare settings.
Okay.
So I think it would be a bit difficult
Okay.
for just having the
Logistics required are different.
Yes.
I mean, maybe permission
So
from
we
the
would
government or
need-
something.
Yes, yes, that,
Okay.
and we will also like to collaborate with hospitals for
Okay,
that.
so the B2B model hai.
Yeah, right.
Okay, so Rinki, now I think more or less we have covered everything. Tell me, have I missed anything which is still not asked to you and you want to tell that? मेरे से कुछ मिस हुआ हो, questions या कुछ ऐसा हमने मिस किया हो related to entire this thing.
I don't think so.
We have done that.
Yeah, we have.
So now because this we are publishing it for the industries. Anything you want to tell them, those who are listening to you? Anything you want to tell them? Why you want industry to collaborate with you? Anything you want to tell them?
Okay, so I keep on saying that antimicrobial resistance or antibiotic resistance is such a significant threat to healthcare globally, not just in a prescribed setting or something like that. So it is a huge problem, and like the World Health Organization is also deemed these pathogens which we are working on, escape pathogens, basically these as critical priority pathogens. So urgent actions are needed to tackle that, and persistence is one of the mechanism which can lead to further development of resistance.
Okay.
So we want the existing antibiotics to keep on working, because it is really hard to develop new kinds of antibiotics which takes like 10 to 15 years to develop a new drug, and that's why pharma industries are also just take a setback,
Trying to-
because it's a long time, too much of manpower plus funds, too much of it is required. So everyone is basically afraid of that, so that we don't know what would be the output of it. So to keep doing that, we want the already approved drug to keep on working.
Okay.
So this is one of the way that we can tackle that problem.
Wonderful. So it was very nicely explained, both of you. I am really thankful to you for that. So I am really thankful to both of you. Thank you and all the best.